Thanks, Walt. Thanks for inviting me. It's a great introduction. I thought that since this is sort of like the first lecture for the session, that I would start off by giving kind of like a brief introduction to mass spec in case there are some like mass spec curious people that have just wandered into the audience. And then I was going to spend the entire time talking about machine learning methods for proteomics, in particular, how fragmentation and retention time prediction has kind of reshaped how we are doing data analysis and thinking about analyzing our type of data. Specifically, I'll talk about the Prosit tool, which was sort of revolutionary for us, but there are several other tools that are in this sort of arena. And I'm going to talk some about ways in which we've used other types of computational techniques to take advantage of those types of tools. And then at the end, I'm going to talk about a little story around a new tool that we've been developing to do additional types of predictions for peptide properties. So just to kind of get everyone on the same page, proteomics is a method to measure peptides as a proxy for proteins. So we don't actually measure proteins directly, except we will see some discussion of that actually later on in the session. But in general, we're measuring proteins that have been linearized, cut with digestion enzymes, and then categorically measured. So where we're trying to monitor for specific proteins that are generated by specific genes in a sort of a sea of other proteins that are generated by those cells. So this issue of trying to separate out the proteins that we're interested in from the proteins that we're not is kind of fundamental to our science. So kind of like the most common technique for dealing with this is to front load all of our mass spectrometry analysis with an HPLC to separate out peptides. So we'll separate them out over time, and we might get a bolus of peptides that have a similar characteristics, similar physiochemical properties that elute at the same time. These will measure at the same time and then kind of gate on a specific peptide based on their mass. We'll fragment these in another part of the mass spectrometer, and then we'll analyze those fragments. And so almost all of the time we're analyzing sort of spectra combined of these fragments associated with a particular peptide. So to kind of put this another way, if I show this plot of peptides eluting off over time with different precursor masses, the most common way to analyze this type of data is with a method called data-dependent acquisition. That's where the data defines the acquisition. So as we are marching through in time space, we're choosing the most abundant peak to go off and try to sequence. So we'll select this particular ion for collecting a mass spectrum with a particular precursor mass, and then we'll stick it on an exclusion list so that we don't have to look at it again. We'll do it for the next one and the next one and the next one and the next one and the next one over time to kind of get an idea as to what is representative of those peptides. But kind of like the big challenge of this type of approach is then trying to reconstruct what peptides those spectra actually belong to. And so that is, I think, something that will drive a lot of the talks today about trying to reconstruct from these spectra what peptides are actually present in our samples. So one way to think of this is try to reverse the problem, right? If we had a peptide sequence, could we come up with a spectrum? And that would make it a lot easier to interpret which is the right peptide spectrum match in this case. So one way to do this was a method that was developed by the Wilhelm group in Munich, where they've developed this method called Prosit, which is a tool that we use quite a lot in the lab, actually, where you take peptide sequences and precursor charges and collision energy settings in the mass spectrometer, and you feed them into a deep learning neural network, and then it produces spectra for those peptides. And they've built two networks, one for predicting the fragmentation patterns for these peptides, and another network for predicting the retention times for these peptides. And these peptide predictions are like strikingly accurate, right? So, for example, this is just a single case that we were using in a manuscript where we were looking at the empirical versus the predicted fragmentation patterns as a butterfly plot. So the top is the empirical data, and then the bottom is the intensities for the predicted data, and they're extremely precise in terms of their estimates, which is kind of exciting. Similarly, if I just kind of look at this globally, right, with the Pearson's correlation coefficient between those two values, predominantly we're getting very high correlation between our predictions and what we actually measure. Not entirely, but in general, it's pretty good. Similarly, the retention time accuracy is not bad, but it is maybe not as precise as the fragmentation pattern selections. So this is an extremely powerful technique that we've been using to start off in a bunch of different ways. And so I want to talk to you about two different programs that we've developed in the lab as ways of trying to use deep learning based predictions for informing our proteomics data. So one of them is an application for DDA using a method called Scribe. This is a new software tool that we've been working on. And then a second tool for doing this similar kind of approach using DIA data, or data independent acquisition data, which I'll introduce again a little bit later. So again, the biggest problem for us is trying to take spectra and match them to peptides and try to ascribe what the actual sequence of that peptide was based off of the spectrum that's present. So we'll take spectral raw files, and then simultaneously we'll take FASTA databases of proteins that we've cataloged, either using DNA or RNA-seq on new samples, or on just analyzing well-known canonical genomes. And so we'll predict entire libraries of all possible peptides that FASTA could produce. So +1 to +4s, or up to +5s, sometimes even PTMs in the cases where we have access to the PTMs. And we stick those into this program, which generates a giant feature file based off of all of those peptide spectrum matches, which get put into a tool developed by Bill Noble's lab, followed on by Lucas Shaw, called Percolator, for determining which of those features are the most important and sort of structuring a support vector machine around choosing which of those features are appropriate. We have a module that does retention time refinement, so it uses the retention time information that's generated by the Prosit predictions, and then it produces quantitation reports or libraries for further use. So to kind of give you an idea for how well these things perform, so just as a benchmark, I'm using MS-Fragger as an analysis tool. In this particular case, we're analyzing a HeLa genome, and this is just the number of unique peptide sequences detected by each of these methods. So if we're using traditional library search tools, like for example, Spectrast and a DDA library, typically we haven't always been doing as well as database searching in general, which is one of the reasons why library searching hasn't sort of taken over the world, even though we actually have very, very good libraries. We just haven't had a lot of very good tools for doing this type of analysis. And so a lot of these tools are based off of simple scoring metrics like dot products and that sort of thing. And so what we ended up trying to do was take some of the knowledge that we had learned from matching kind of like not perfect spectra using DIA methods and not perfect predictions, and try to come up with a scoring engine that was able to deal with the inconsistencies in the predictions and in the data. And as a result, we've developed this tool that does not just better than Spectrast, but also better than sort of state-of-the-art DDA search engines. One of the things that's sort of interesting is that this is just with a DDA-based library. Our goal really wasn't to do it with prediction, with DDA libraries. Our goal was to do it with predictions of entire FASTAs. And I think one of the things that we've shown here is that while the search space gets far too large for other types of search engines to be able to make use of it, the sort of coupling of Scribe with Percolator has given us the ability to detect more peptides than we would have if we were searching these smaller, more constrained libraries that typically you would have searched with in the past using other types of tools. So, so to kind of give you an idea as to how consistent these measurements are, here are the peptide overlaps between triplicate analyses done by MS-Fragger and Scribe and Spectrast, where the size of the Venn or the Euler diagram is proportional to the actual number of peptides detected. So what we're finding is that even just within the overlap of the three the three detections or the three replicates, we're making about the same number of detections as we were with any one replicate just in total from MS-Fragger. So the results are not just we're getting more detections, but they're also more consistent. And in particular, if we compare the sort of peptide overlap of detected peptides that we're finding from Spectrast, this sort of straw man representative tool, versus Scribe, they're almost entirely contained within each, or Spectrast is almost entirely contained within Scribe. Similarly, MS-Fragger actually detects some other peptides, but already half of them are from PTMs, which were something that we didn't actually include in the search space for Scribe to begin with. So we think that the ability to use, and it's completely logical, right? The ability to use fragmentation patterns, the ability to use retention times is just better than not having access to those predictions to begin with. So it seems sensible that we would be able to make these bigger detections. This is much more important when we're starting to talk about other types of peptides, though. So for example, just as a comparison, here is a data set of HCD collected tryptic peptides and CAD collected tryptic peptides, and we're getting 30 to low 40s in terms of the number of percent improvement over MS-Fragger. But if we start looking at some of these non-tryptic peptides generated by other digestion enzymes that don't fragment quite as well, here having access to accurate fragmentation patterns becomes much more important. And in this particular case, despite the fact that Prosit really wasn't trained on non-tryptic peptides, it actually generalizes quite well to non-tryptic peptides, we found. And so it becomes a really powerful tool to be able to identify peptides that are difficult historically to make measurements of. Okay, so as I was saying, this is our Scribe tool, but in general, we're actually not using it that much in our lab at least, for doing detection off of DDA data, because in general, we're not really collecting a lot of DDA data in my lab. So a lot of the focus has been on using Scribe as a library generation tool for other types of techniques, for feeding into DIA-based analysis or data independent-based analysis or targeted-based analysis, so using Skyline. So these kind of library generation tools have become sort of the bread and butter of this in our workflows. And I want to talk to you about how deep learning has kind of changed how we've been analyzing DIA data too, so from the same framework. So just to kind of give you some context again, here is the sort of DDA framework where we're selecting particular precursor masses and charges for sequencing with the mass spectrum. So with DIA, we actually do a slightly different type of measurement, where instead of fragmenting each peptide individually, we're actively multiplexing our measurements. We're actively co-fragmenting multiple peptides at the same time. So instead of selecting out 507.4, we're selecting out a range, 500 to 525, for example. And we're constantly co-fragmenting those across time. So seeing the fragmentation over time. And as a result, the actual fragmentation patterns at any one of these individual spectra are extremely complex. They're a mixture of peptides of multiple types with multiple different precursors. And so a lot of the sort of trick in trying to analyze this data is trying to pick out which of those fragments actually coalute with each other and belong to the peptide in a sea of other peptides and a sea of other fragmentation patterns. I think in some ways this is a really powerful technique because many of these sort of peptides that would be potentially lower abundance would have been completely missed because they wouldn't have been selected because their intensities wouldn't have been high enough for measurement with data-dependent acquisition. But with DIA, the problem is now on the computational shoulders, right, where we have to extract out the signal of this low abundant peptide from this sea of otherwise noise to be able to interpret that detection. Okay, so here is a field where in trying to do this, library searching has already become, sort of the most common practice, right? There are methods to be able to detect peptides directly from these types of these types of data sets, but if they don't use deep learning themselves, they're usually poor performing tools. So just to kind of give you a comparison, one of those tools that we developed is called Pecan. And so this is the number of detections that we see. And with this tool, and Pecan just sort of stands for peptide-centric analysis of DIA data. And so it's just saying, okay, for all of these B and Y ions that I expect to see for a given peptide, I don't know anything about their fragmentation. I'm just going to extract them all and see where the signal kind of coalesces, where we start to see peaks. And that does detect peptides in a lot of these types of samples, which is great, but it definitely is underperforms versus sort of the most common approach, the state of the art, which would be to use a DDA-based library using a tool like Scribe to be able to aggregate a bunch of DDA data together to tell us, what the good fragmentation patterns were, what the good retention time patterns were for each of these different peptides. Okay, so in this particular case, I've aggregated, and this is important, right, where we're doing SCX and high pH reverse phase, two different types of separation techniques to be able to identify as many peptides as possible. Here, for a yeast proteome, we're covering that in about 16 different injections. For a HeLa proteome, we're covering that in 39 different injections of the same sets of samples, so fractionated and then analyzed independently. So if we analyze this type of DIA data using library searching techniques, again, using kind of Prosit predicted libraries, so these large libraries, we're oftentimes still detecting fewer peptides with our predictions than with our DDA-based libraries. And the reason why we see this drop is oftentimes we think is because of search space considerations, where libraries are just significantly smaller in scope, or these DDA-based libraries are smaller in scope. They only contain peptides that have already been observed before. And so when we search these predicted libraries, we have to search for all different possible peptides from every possible protein, not just the ones that have been in proteins that have been observed in that sample, but also from peptides that have been observed in those samples. Okay, so to kind of combat this, we've tried to develop other ways of extracting this type of information out. And one of our approaches has been to segment our DIA-based acquisition into two different frameworks. One is to acquire quantitative data, and the other is to acquire data that is specifically designed around detecting peptides, rather than just quantifying peptides and detecting them in the same runs. So, for example, for this type of method, we might take a data set where we're analyzing from 400 to 1,000 MZ. So this would be all the peptides between 400 and 1,000 MZ in these 24 MZ wide windows, 24 MZ wide windows. Just like how we were talking about before, these are wide windows with a lot of co-fragmentation. We have been acquiring this data, or at least of a pool of these samples, using a very sort of expensive approach from a measurement perspective, but we're doing this of a pool, so where we're actually just trying to detect all of the possible peptides in our sample. So we'll run one acquisition from 400 to 500 in 4 MZ wide windows. We have a technique to sort of stagger these windows in a way that we can deconvolve them, which gets us down to 2 MZ wide windows, which gets us essentially the same quality data that we would get from DDA, or really the same quality data that we would get from targeted PRM style acquisition, but for all of possible peptides in that proteome. So we'll make that measurement in one acquisition, and in the second acquisition, we'll measure them from 500 to 600, 600 to 700. And this is really expensive if we wanted to do this for every one of our quantitative samples. But if we just do this of a pool, it gives us kind of the ability to say, this is what our comprehensive proteome is, and use that to make detections of going forward. So just to kind of put this in another framework, like if we're looking at DIA this way, where we're co-fragmenting a bunch of possible peptides over some retention time with different sort of precursor mass windows, here we're dividing the precursor mass, or the M over Z space, into very small 2 MZ wide windows that can essentially isolate as if we were targeting those peptides. Except instead of targeting specific peptides, we're targeting all possible peptides across all of these different windows simultaneously. Okay, so our workflow for trying to do this analysis is we'll take FASTA databases and we'll generate an entire predicted library, just how we were doing with Scribe. So this would be a list of all possible peptides that could have come from any of those different proteins. And then we'll take our pool sample and we'll inject it again 6 times with this detection-focused measurement. So 2MZ wide windows, so 6 injections, and then we'll do that sort of analysis where we'll try to identify these predicted matches versus our gas phase DIA matches. And again, as I was saying before, their fragmentation patterns aren't going to be identical. They're going to be mostly the same, but they won't be perfect. The retention time patterns are going to have a little bit of scatter around them, just like how we were showing before, same as before. But rather than try to do that for quantitation for all of our samples, we're actually generating what we call an empirically corrected library. That's where we're taking it, rather than using the predictions, we're taking the empirical data that we've generated, that we've matched to those predictions. So we'll take those gas phase DIA fragmentation patterns down here and the actual retention times for the peptides that we detect, and we'll stick those into a library, and it's limited in scope, so it's only the peptides that have been detected in that pool of samples from your experiment. And then from this, we'll take all of our quantitative samples, which we'll measure in individual normal DIA runs, and then match them up. But one advantage here is that because all of the fragmentation patterns are done on the same mass spectrometer at the same time in the same background, they're almost identical in terms of their fragmentation patterns. And similarly, their retention time patterns, because it's acquired, because we're essentially matching the retention time patterns in wide window data to essentially data that we collected using narrow window data, the retention times are exactly the same because they were measured on our instrument at that same time. So this is a way that we can actually take advantage of these predictions, where we'll do sort of a hybrid approach where we mix sort of data acquisition and predictions to be able to generate these empirically corrected libraries where they're honed specifically for our instrument. And we find that we produce libraries that give us significantly more detections. So not just fractionally more detections, but significantly more detections versus just analyzing a traditional DDA-based library for DIA analysis. So this is a super powerful technique, we think, for analyzing DIA measurements. And it's true that these Prosit predictions can be sort of strikingly accurate, but the accuracy of these can be difficult to measure for all peptides. So there are some peptides, for example, down here where we don't actually get very good. I don't know if this is visible. You guys can't even see my mouse. Okay, never mind. There are some. I know that the people in the audience outside can't see this, but there are a bunch of peptides down here, right, that you can't see that have bad like retention times that don't match the predictions. And then similarly, there are some peptides over here that don't have retention times that match the predictions. In general, we find the fragmentation patterns are pretty accurate for most types of peptides, but the retention time patterns could actually be, there could be some improvements. And one of the reasons why we've been going down this road is for trying to measure PTM. So post-translational modifications are oftentimes found with multiple different positional isomers. So for example, in CDK1, there are these 2 different positional isomers where you can phosphorylate this peptide at this threonine or this tyrosine, and they're right next to each other in terms of time. They have exactly the same precursor mass. And they have most of the same fragment ions. So the only fragment ions that differ are these 2 fragment ions, or +1 fragment ions that differ are these 2 fragment ions that you could measure. So this becomes a continual challenge for trying to analyze phosphorylated samples and in this particular case, phosphorylated samples, but all types of PTMs using DIA-based methods, but also DDA-based methods, because they have very similar characteristics chemically and structurally. And so this is continually challenging, because both of these sites being phosphorylated at either one of these essentially has the same sort of function downstream, but it definitely indicates different biology for who phosphorylated whom in this circumstance. So we'll get essentially the same, exactly the same precursor masses. Most of the fragment ions are the same, but the HPLC traces might actually differ a little bit. Oftentimes we'll see phosphorylation peptides separated, phosphorylated peptides of different positional isomers, separate by about a minute or two, or maybe a half a minute. And so if we can develop resolution that can sort of analyze this, then it can be very powerful for knowing what's going on. But otherwise, we're kind of lost in trying to analyze a sample like this. For example, this is a measurement where if you were to make a call based on the fragment ions that are measured here, you would say this is a very logical peptide. But if we look at a heavy standard, we can see that we would be measuring the wrong positional isomer. And this just happens to be a certain case where we have a lot of the same fragment ions are being found in both of those circumstances. So the best tool in our arsenal for a lot of these cases is very, very accurate retention times. So that said, we have a little bit of a headway here, where if we actually monitor retention times that have been sort of crudely sort of aggregated using IRT peptides, or a handful of like 10 peptides to align a bunch of runs, if we look at Prosit predictions, they do a bit better than those kind of crude retention time alignment methods. But if we want to sort of measure the same sample just using a different column, we get sort of even better retention time accuracy. The difference between the Prosit predictions and this sort of same sample different column indicates this is sort of like the range of where we can optimize, I think, the amount of headroom that we can get on top of these peptides. Okay. So with that, I want to just launch into a quick story around a tool that we've been trying to work on called Chronologer, that is focused around making more accurate retention time predictions than sort of the current state-of-the-art tools with deep learning. And a lot of this follows around this idea of deep learning being kind of like a game of telephone, right? So where you have some information that has been derived from a peptide sequence. So you have this peptide sequence that you kind of structure in a certain way, and you feed it into the first layer in the network, and that kind of makes an interpretation, and then you feed that interpretation into the next layer of the network, and then you make an interpretation of an interpretation, and then an interpretation of an interpretation of an interpretation, and so on. It's kind of like a game of telephone where you're sort of slowly losing the concept of reality as you're feeding information from one layer of the network to the next. So we find that we've been exploring different newer types of network topologies that seem to kind of get around sort of these problems. So Chronologer is a tool that uses a different type of technology than an LSTM. This is using a ResNet-based architecture, and the way in which this is designed is there's an embedding layer, and there are a bunch of these sort of network blocks, residual network blocks. And so the idea here is, in this case, these are dilated. So each of these blocks that we've got here sort of increases the space or the scope around the number of amino acids that we're considering at each one of those blocks to give us a little bit new information for each one of them. And that each one of these blocks has a skip function where it passes information from the beginning of the network. If it decides, or if it needs to, it passes some information to the end of that block so that it can feed forward what was essentially the original state of that network. So in this particular case, there are really only 6 convolutional layers, right? So there's an embedding layer, 6 convolutional layers, and then some like interpretation layers essentially, and then a dropout layer to sort of make sure that everything is well controlled. So so there are very few parameters in this network, actually significantly fewer than there are in Prosit or in other types of tools like DeepLC from Leonard Martin's group. And we find that that even with this simpler network with fewer parameters, we actually can achieve using the same training data from other tools. So this is the training data from Prosit, which achieves a mean absolute error of about 1.25. In this particular case, this is minutes on real retention time. So this would be the error rate for that. Chronologer achieves maybe about a 20% reduction in that mean absolute error. And this holds for sort of all of the really high quality, I think, learning tools out there right now. So DeepLC is a very powerful tool that is similar in scope to Prosit with some additional learning features. And again, we're still achieving that same pretty much 80% mean absolute error versus the predictions. Again, compared against an unpublished SSR calc, this sort of the most modern version of SSR calc from Ole Kronken's group. This gave us a lot of hope that we were onto something. But the other thing that gave us a little inkling of inspiration was the fact that these data sets that these guys were trained off of, even though, so in these cases, we're training off of exactly the same data as DeepLC used or as Prosit used or as SSR calc used. We're achieving better results. In this case, if we look at actually the overlap in those peptide sequences, there's shockingly little overlap because of how they were derived, right? So the data set that was being used by DeepLC was predominantly generated by DIA data sets of human and predominantly human samples. The Prosit predictions were based off of proteome tools, which are all synthetic peptides. Kronken's data is based off of very different, not synthetic peptides, but other types of organisms, non-human sources. And so if you were trying to aggregate all this data together, the sort of most common way would be, say, there's a certain number of peptides that are in common between all of those data sets. And you could use those as a retention time anchors and map from one library space into the other library space to give you an idea as to what library B would have looked like in library A's retention time space, and then create this combined library and retention time in the A's retention time space. But if you're trying to do this for a lot of data sets, and we've been trying to scavenge some of the best data sets we've been able to find across the community, here are 11 data sets that are in our, were part of our interest. There are very few peptides that actually are in common between these. So for example, the vast majority, almost tenfold or over tenfold, are actually only in one of these samples. And there is no peptide that is shared amongst even 7 of these different samples or sample sets. So there was no way to aggregate these data using kind of the common IRT-based approaches. So again, we looked to our old friend Prosit for some advice here, where we were looking for all of the possible peptides that would be Prosit predictable. These would be peptides between 7 and 30 amino acids that would have reasonable mass and characteristics and be unmodified, right? So that was another key feature. And so we would use these pool of Prosit predicted peptides to be able to put them not on a line with another data set, but actually in what we call Prosit IRT space, or the retention times that Prosit makes predictions of. And so basically it lets us map these 2 disparate libraries A and libraries B both into Prosit IRT space instead of into an actual real IRT space or real retention time space. And all of these data sets have at least some peptides that correspond to these. Even the phosphopeptide data sets have some peptides that kind of escape that system. Even the TMT data sets, we think of that reaction as being a perfect reaction. It always goes to completion. It doesn't always go to completion. We can definitely find unmodified peptides in those data sets. And we constructed this retention time library database. It was combining all of these different data sets together into 2.4 million unique peptide sequences. To give you context, a massive KB doesn't contain retention times, but even if it did, its size is only 1.1 million peptides. So it's as if we had collected 2 massive KB sized libraries together. So using this this framework, we're actually able to make very accurate predictions of these peptides. So in these particular cases, we're seeing about 60% reduction in mean absolute error relative. And so if I put these on a straight line, for example, you can see that the purple on the bottom, the error of the purple trace is much smaller than the error of the blue trace like DeepLC or SSR calc, the red trace. that this is a very effective tool for not just tryptic peptides, but because we are built this library off of non-tryptic peptides as well, it performs very well for non-tryptic peptides too. So an improvement in accuracy versus DeepLC and Prosit in those cases. Similarly, for column temperature, it doesn't have sort of bias for one column temperature or another because it's not built off of only one data set. And then similarly, it performs equally well with phosphopeptides. The same single model actually makes these predictions because we aggregated all these data sets into the same space. So you can predict phosphopeptides and non-phosphopeptides using exactly the same model, as well as the other 11 different classes of PTMs that we've developed for our model. There's a retraining system where you can add information in, but rather than sort of retrain the model, we augment the current model. So we add in information to that model so that we can keep the training accuracy of all of the other peptides as well. So going back to this plot of accuracy, where we're looking at sort of the pan-human IRT set and sort of the optimal, this is kind of representative of perfect prediction and Prosit, we feel like we're actually pretty close to this. So these are actual measurements of technical replicates of different columns of the same samples versus the Chronologer output. And I think one important characteristic is no data has been actually used for this system. No training data in this plot has been used for our predictions. So just to kind of wrap up, peptide predictions we think have a very powerful use, or very powerful application for both DIA and DDA-based data sets. Changing how we collect data. So that means changing the sort of data that we're acquiring for identification purposes versus quantitation purposes can make bigger gains than actually just improving only the computational methods. And then this last story about using current generation tools to basically curate data sets for our newer tools, I think gives us the ability to make much more accurate, much more useful, big library sets for us to be building these deep learning models off of. So with that, I'd just like to thank the collaborators here. So Damien Wilburn was in my lab until quite recently. In a month, he's actually transferring out to start his own lab at Ohio State University as well. So we have a great team here at Ohio State. Damien did all of the work with the peptide retention time predictions, along with help from Daniel Swaney's lab and Oleg Kronkin's lab. And that a lot of the Prosit prediction work was done in collaboration with Matthias's group at Munich. So with that, I'd just like to say we're hiring people to do work like this, and we're excited if there are any people that are excited in this type of work. Okay, great, thanks.
I'll just talk that. What led you to, I often run into this question myself, what led you to choose a convolutional architecture as opposed to a recurrent, especially for 2D data?
That's a great question. And we explored a bunch of these different types of architectures to make decisions on it. Here we were interested in being able to, so one of the issues with this type of data is that you want to essentially look at adjacency, right? And how much do adjacent amino acids affect other amino acids? And we find that that is the most important characteristic, and that's why essentially having that convolution slide across is the best way to think of it. And that's sort of technology that was well described in the Prosit paper, and we think actually does make a lot of sense. But that was our rationale behind making that choice. There are other ways that you could try to think of this data as well, using transformers. We find that our approach sort of like is a nice middle ground for that. Again, our goal was to make the simplest network possible, actually, rather than make a very complicated network where we had lots of different types of inputs and lots of different type of manipulations. We don't want to, we're not smarter than the machine in this case, right? The machine is better at figuring out different characteristics of these things. So we actually there are only really like 6 tunable parameters in our model in terms of things that you have access to tune, or that different parameters that are available.